Individually speaking, they are 46. If you count them 1, 2, 3, 4 like that. From the pairs, pair means 2. Okay. So for this, we will conclude this topic about the cell, the nucleus, and the chromosome. Now, each time the cell divides, the same exact number of copies of chromosomes must also be replicated in every cell, because you see, we cannot have a different kind of a genome in our body. So whatever somatic cells, the body cells that we have, must replicate exactly the same, otherwise it will mismatch. So that is the most important aspect when it comes to replication. Making more cells of the same kind. Now, in order for that to happen, even your DNA must have to divide, you see, must have to replicate. So, this kind of cell division is called as mitosis. Mitosis. My means dinuclei, meaning the cell divides into two nuclei. So, supposing if this is a cell, now this cell has to transfer all its genetic material, which is the chromosomes, whatever inside the contents, must duplicate exactly the same in these two cells. So, in short, whatever chromosomes that have existed here must also exist here. Whatever has to come here also must be here. Understand? Whatever the chromosomes has to come here must also be there, So that is the fundamental concept. In order for getting these chromosomes into this cell and into this cell, each of the chromosome must also, which has DNA in it, must also divide into two. This is what we call it as DNA replication. So inside each of this chromosome, we have got the genetic material, which is the DNA. So now if you see the karyotype, which means this one will have its DNA, and that DNA must replicate into two forks. The same way, this also must replicate into two forks. The same way, even this one must replicate into two forks. This one must replicate. So this is what happens. So when we talk about mitosis, when we talk about mitosis, we will go into this, how the cell exactly divides, and the cell division, we will go into those aspects in detail. Okay? There are what is called as interphase, prophase, metaphase, and there's a lot of stages that goes in the mitotic cycle. Understand? Mitotic process of cell division. Understand? There's another one which is called as meiosis. This is mainly for sex chromosomes, which is for the sperm and also for making the egg. Understand? So that is how meiosis is. Mitosis is basically for somatic cells. Those are mainly for germline cells, which are for sex cells. So with that as a background, we will go into what is called as DNA replication. This is one of the most important concepts that you need to keep in mind when it comes to DNA, because there is another process which is totally different but also starting from the same DNA, that is called as protein synthesis. Protein synthesis we will discuss in another lecture. For now, we will discuss about DNA replication. Now, in order to understand DNA replication, there are a few terminologies that you need to keep in mind. These terminologies will help you understand the concept of DNA replication. So what happens in DNA replication is a single DNA will unzip. In order to form two exact copies of the same DNA. Now this process of unzipping and then adding new nucleotides into each of the daughter DNAs, each of the DNAs which are splitting out, that process will be the DNA replication. So, in order to understand DNA replication, I will tabulate just a few important terminologies and their functions which you need to understand and know. The first one, we will call it as helicase. Helicase is an unwinder. For example, the tape recorders or whatever you have, it has to wind and unwind. So this is more like an unwinders. Okay, so I put it as unwinder. Some people call it as unzipper. So basically, what it is, let me just explain to you. Supposing if this is the helix. Now this has to first unwind. You see, it has to unwind and then it has to unzip. You see, so the helix needs to unwind and the helix needs to unzip. So that's the key point when it comes to the role of helicase. So helicase is basically going to unwind and unzip. So I just put two lines like this, which means this has been unzipped. Okay, so that's the key point which you need to know. This is one of the important important role in the context of DNA replication. Now, after it has unzipped, the next most important thing is that is what is called as primase. Primase is more like a gluer. Primase is the initiator. Sorry, not the gluer. Primase is the initiator. Initiator. In order for the unzipping to take place, so something has to initiate to start the process. So primase is the initiator. Number 3, ligase. Ligase is the gluer, like a glue to fill up some of the open pockets. So ligase is more like a gluer. And one of the most important thing in the context of DNA replication is you should know the role of polymerase. So number 4. See, for example, in the context of protein synthesis, we have got ribosomes. That is a key component in protein synthesis. In the context of DNA replication, the most important player in this drama is the polymerase. In polymerase, there are two types, type 3 and type 1. Type 3, we'll call it as the builder. And type 1 is like a building inspector. After the process has been done, it will proof check. It will go and check back and everything is okay. So this is a building inspector. So if you familiarize yourself with these terminologies, helicase, primase, ligase and the polymerase, then your understanding of DNA replication will be very familiar. So let me start with this. Earlier on, we have discussed in detail what is a 3' end and what is a 5' end. Just to familiarize yourself very quickly, I will go through the 5' and the 3'. We know that there are 3 most essential components that are of vital interest in the context of DNA and in the context of RNA. One is the phosphate molecule. Then you need the sugar. And then you need the nitrogenous base. So this is the base, this is the sugar, and this is the phosphate. We'll put it as P. So this is oxygen, This is carbon number 1. So I just put C1. We go clockwise. C2, C3, C4. And then here we've got another carbon which is C5. So in the context of DNA, which is deoxyribonucleic acid, here we have got OH, but now here the O is missing. Is it correct? Okay, that's why we call it as deoxyribo. There is no oxygen is lacking in one of the carbon. Okay, so just to familiarize yourself, and these phosphate and the sugar will form the backbone of the DNA structure. Understand? Now, why I do this is I wanted you to know what a 5 prime and what 3 prime is. Here you see 5, this is 5 prime, and this is 3 prime. Understand? So you can see it is 5 prime to 3 prime. Understand? Now the opposite, the corresponding nitrogenous base will form a hydrogen bond with this hydrogen bond with this. So there is not much space for me to draw, but you just have to draw exactly the same thing, but just in the opposite direction. You see, just in the opposite direction, which means you will have the oxygen at the bottom. I'm sorry, let me put it this way. Okay, oxygen at the bottom. So the sense of direction changes. Understand? This is what we call it as anti-parallel, because from here it's going from 5 prime to 3 prime in this direction, and in this you see 5 prime to 3 prime going just in the opposite direction. So they are called as anti-parallel. Understand? So I just wanted to briefly introduce you to what 5 prime and what 3 prime is. So now the next one will be the same, similar like this. So you just need to keep building on the phosphate and sugar, and this will form 5 prime, 5 prime, 5 prime, like this, 5 prime to 3 prime. So this will be the direction. You just have to keep adding more of this sugar, nitrogenous base, everything. And I'm sure everybody is familiar with the base pairing rule. We know that adenine, guanine, thymine, and cytosine. A binds with T, G binds with C. So this is the base pairing rules. So two of them together we call them as base pairs. Understand? Individually we call it as a base. So with this as a background, we will go into what DNA replication is. Again, I would like to emphasize and repeat. The players in this drama are helicase, primase, ligase, and polymerase. Polymerase 3 and polymerase 1. Now you should not ask me a question, why not polymerase 2? And why it is polymerase 1 and 3? These are some of the terminologies which scientists have given. So those we have accepted it as it is, and we need to know it the way it is. So now, having known what 5 prime, what 3 prime end is, you should understand that 5 prime will only bond with a 3 prime and vice versa. Understand? So with this as the background, we will enter into this critical stage in cell replication. In order to know that, we should know what DNA replication is. And you know very well that this DNA is coiled on a protein called as histone, and it is compressed inside the chromosome. So that is what we call it as a chromosome. In fact, chromosome is nothing but a DNA, but which has been highly compressed. So with this as the background, I will draw the DNA in a linear fashion so that we will also get the concept right. Now, we are going to draw a simple structure of a DNA. For, as I told you, DNA is always right hand helix. You see, it has to go right hand. There is nothing like left hand. Left hand, if somebody draws a DNA on the left hand, which means it's wrong. So DNA has to have a right hand spiral or a helix, if you would like to call it as helix. So now, Let me draw a simple structure of DNA, something like this. You might have come across this wherever you have read about DNA. You might have come across this. You should understand one of these end is a 5 prime, and then if it goes like this one will be a 3 prime, and this also will be a 3 prime, and then if you go like this one is a 5 prime. Now the first stage is to unwind. So when you unwind, it will be like this. Understand? And this one will be like this. So here we will draw it as a 5 prime, and here we'll put it as a 3 prime. So here it has to be 3 prime, and this has to be 5 prime. And you already know very well. See, I have not drawn those nitrogenous bases which are corresponding here. So you see, these are those nitrogenous bases, and in between you have the hydrogen bond. Hydrogen bond. Okay, and this backbone is a very strong bond. These are covalent bond, also sometimes called as diester bond. Anyway, so these are the backbone. The backbone does not change. What changes is only the base pairing. Understand? If it is this one is A binds with T, so like that. Okay. So now, for the sake of simplicity, assume now the unwinding process has taken place. So now it's basically a ladder. Now, basically a ladder in this fashion. So you can come up with your own combinations. This is A, this is T, and U, this is G. So you can just come up with your own understanding of the nitrogenous bases. Now, what happens is, first, this enzyme helicase has done the unwinding, and now the unzipping. Unzipping means you are going to now unzip in this fashion. Okay, we are going to unzip in this fashion. So what happens here is this one has unzipped. Okay, so I just put a direction like this. So it's unwinding and unzipping. Understand? So the same thing also happens here. Unwinding and unzipping process happens. Mind you, this is a very fast and a very quick process. So we are just trying to show you exactly in a simple way, but actually this process takes place in around 50 base pairs per second. So that's the high speed rate of addition of base pairs onto these DNA strands. So now the unzipping process has happened. Who does the unzipping process? Helicase. Helicase enzyme. Enzyme helicase does this unzipping process. Now, let me rub this top portion over here, and then we will go more into the details of DNA replication. So we have seen 5 prime to 3 prime. That's the direction we have written. So we will maintain the same direction in our explanation as well. So here we go. So this is the unzipping, the first one. And this is how it is. 5 prime to 3 prime. And here this side we have the 3 prime and the 5 prime. Now what happens is, there has to be an initiator. See, in order for something to start, you need to initiate. You need to initiate. That initiator is called as a primase. So I'm just putting a little bit of a start, like a kick start. So this primase lays down a small piece of not DNA but RNA. You see? RNA. This we call it as a primer. Primer. It's more like a paint, you know. Before you do the final painting, you put a little bit of primer, something like that. So you can imagine like that. So this is the small piece of primase. Now, We know that DNA replication occurs from what end? 5 prime to 3 prime. So this is the 5 prime, isn't it? This is the 5 prime. So now, can you add nucleotides here, or you can add nucleotides here? Where can you add the nucleotides? I mean, the new nucleotides which are coming up. Where can you add? You have to add it over here, isn't it?
Yes.
So it's starting from the 5 prime to the 3 prime. Now, can you add nucleotides here? You cannot, because the DNA replication happens only starting from the 5 to the 3. So that is why we cannot add nucleotides on this end. I hope the concept is clear. Are you with me? Yes. So this little small piece of layer is called the primer. It's laid down by the primase, and it is not a DNA, mind you, it's an RNA. So here there might be a little bit of a confusion. We are going to see how this is going to be sorted out later on. So now, here comes the builder. Builder is the one who is going to put new nucleotides. Are you with me? Okay. Now you know already. When you unzip, you already have one end of the base pairs over here, isn't it? Okay. So you might have adenine, guanine, cytosine, thymine. The opposite of this is over here, isn't it? So the one which is going to pair with this is thymine. For pairing with G is cytosine. Here is guanine. This is adenine. Just for the sake of simplicity. But you see, the one who initiates everything is the primase, which is a small piece of RNA. So this is the one which kickstarts the whole process. Now, as we discussed, the DNA replication happens from 5 prime to the 3 prime end. Now, who is the builder? The builder is polymerase 3. So I'll put it as Pol 3. That's the builder. Polymerase 3 comes along and starts laying down the new nucleotides. I mean, the new bases, rather. The new bases. Is that clear? Polymerase comes and starts to add the new bases over here. So now what happens is, you get the opposite of A, which is T, G, C, and C binds with G. So anyway, so here you are getting the new nucleotide bases. So that's what happens in this region. Is it clear now? So we have unzipped. This region is now sorted out. So this is 3 prime, and it goes on like that. So DNA replication will always be from 5 prime to 3 prime. So in order for this 5 prime to bond, it has to bond with a 3 prime. So now the problem comes with this. 3 prime is over here. Can we add new DNA bases, the base pairs over here? Is it possible to start from here? Is it possible to start from here? For this one, you cannot do it. So the problem here is you have to go backwards from this end to this end. Okay, so this strand is called as a leading strand, and this strand is called as a lagging strand. The reason why we call it as leading strand and lagging strand is because this one is straightforward, but this one is now 3 prime. You can't put another 5 prime there because it always goes from 5 prime to 3 prime, which means 5 prime is on which side? On this side or this side? 5 prime is on this side. This is the 5 prime. So you can add only from this side to this side. Is this clear? The concept is clear? Okay. Now, there is another little terminology which I would like to add in the context of DNA replication. When this fork splits, you need some kind of stiffeners. You see, some kind of stiffeners. So there are small proteins over here which are being added over here. These black dots. These are called as single-stranded binding proteins. These are called as single-stranded binding proteins. This is the leading strand, this is the lagging strand. These single stranded binding proteins are being laid more like a stiffener. So now they are constructing the first component of the DNA in the context of DNA replication. Now we need to do this as well. That's the key part in this. Now, in order to make it more simpler, let me draw the same diagram a little bigger. Now, this one you have known. This is what we are into. The same thing, I'm just drawing a diagram over here. Now, here we are through. We know what is happening over here. Now, our focus is going to be on the lagging strand. Okay? On the lagging strand. Now, what happens in the lagging strand is you need to go from this end to this end. Instead of the usual procedure of this is 5 prime to 3 prime, which is okay, this is the leading strand. But now here you have the 3 prime end, so you cannot do it. So what happens again? This silly polymerase doesn't know where to start. That's the big problem. You see? Doesn't know where to start. So what happens? Primase comes along and lays a small piece of RNA. So primase, so again, I'm not drawing all those nucleotides, the base pairs, the bases rather. So I'm not putting those, but just for sake of simplicity, I'll put it over here. Now what happens is primase will come and put a very small piece of RNA. Now this is a RNA. That's the thing which primate is. Now polymerase 3, the builder, comes and starts putting the new nucleotides over here. Fantastic. So it starts putting all the new nucleotides over here, which is complementary. Complementary means the opposite. That's why we call it as complementary base pairing. So we even call this as complementary base pairing rule. A binds with T. So that's complementary base pairing. So it has done this. Now we are going to introduce another new term. This little piece of this, including this piece of RNA which is over here, is called as a Okazaki fragment. Now if you notice very carefully, this end is 5 prime, this is 3 prime. But at the end of the day, they have accomplished our job. The lagging strand has already got the new nucleotide bases. So is it clear for now? So this part is clear. Now, this is going to continue. It doesn't stop here. The unzipping will continue, will not stop. So now what I'm going to do now, for the sake of simplicity, I'm going to unzip it further. So what happens is, let me rub this. I'm going to draw this actually again one more time, but now in a bigger fashion, so that now you appreciate the concept of DNA replication. It's not a complicated process or a complicated phenomenon, but you just need to remember some of these terminologies. So now I have unzipped up to here. You can see how far we have unzipped. Okay? Now, I'm not going to put those nucleotides, the nitrogenous bases and all those things on the nucleotides over here, but just for the sake of simplicity. Now, this was 5 prime and this is 3 prime. Do we have any problems adding from 5 prime to 3 prime? No problem. So I'll just, okay, of course, there is this primase has laid a little primer over here, which is a RNA. Mind you, it's not a DNA, which is a RNA. And now we have a complete brand new strand over here, which is complementary to this. This is the leading strand. Okay, this is the parent DNA. Now we have made one daughter DNA. But now this is perfect. 5 prime to 3 prime. There's no problem. The process continues very well. If you look at the previous image, we saw that primase was somewhere here. Primase. let me just put the new strand over here. So primase was somewhere here, and then the replication took place in the opposite direction. This is more like back stitching. You see, here you are stitching correct. Now it's like you have stitched from here. The next stitching will again start from here. That's the concept. Here you have done the stitching. The next stitching will start from here. Okay? So this is the primer laid down by primase. This is primase. Okay? So we know that this is the Okazaki fragment. Okazaki fragment. Okay? This whole section we saw in the previous drawing. This is the Okazaki fragment. If you see, this side is a 5 prime and this side is a 3 prime. So 5 prime to 3 prime. That's what has happened. Exactly what happened here, but it happened in the opposite sense. From in the reverse direction. Now we call it as back stitching. Now, the same question over here, the same problem over here. Exactly the same problem. Again, we need to start building from here to here. We need to build the next segment. On this side, on the leading strand, we have got no problems. But now on the lagging strand, we have got the problem. Again, this silly polymerase does not know where to start from. So again, primase has to put a small piece of RNA. Primase puts a small piece of RNA, and that's when this polymerase 3 comes in to finish up the strand. So this keeps on going and going and going and going, and that's how the DNA replication takes place. Now, your question is this: what happens to this RNA? What happens to this RNA? And now there's another little thing I forgot to mention. There's a slight gap over here. It doesn't go and complete to the RNA. There's a slight gap. So whenever there is a gap, we need to glue it. So there comes the ligase, which is the gluer. So ligase fills the gap. Ligase fills up this gap. So this gap is filled. But we are not yet done. We are again having something to do with this 2 RNA primer. Here I've just put 2, but this process keeps going on and on as it keeps on unzipping, as the helicase keeps on unzipping. So you have seen these 2 RNA. Now the other builder, whom we call as a building inspector, comes over. Now he says, RNA, what are you doing here? You're not supposed to be in a DNA strand. So he comes and gets rid of this and puts up a DNA. And also here it asks, what are you doing here? You're not supposed to be here. Now so it comes and puts up a strand over here. This builder, we call it as polymerase 1. So polymerase 1 replaces the old primers which have been laid down over here by the primase. So that way everything is complete. It is going from 5 prime to 3 prime. So the zipping process keeps on occurring in that way. Now, so far so good. Now there are 2 little things which I want to mention. DNA replication is so accurate, there is hardly any mistake that happens. So now we have got two new DNA, and here again you have got another new DNA. This is brilliant now. So what happens is, this is how your chromosome, which is packed, gets into 2. So one of this will go into one of the daughter cells, one of this will go into the other daughter cell. So that's the basic concept that's why the DNA must replicate exactly the same in somatic cells. When it comes to germ cells, which are gametes, we will discuss about that another time. As far as here, it's almost exactly the same copy which needs to enter each of the two daughter cells. So far, so good, and everything is done. Now, there is an important concept here which is called as proofreading. Proofreading. Proofreading means polymerase is a very powerful enzyme. It's very important and very vital, this build a polymerase. It's able to go back and do any kind of corrections in the base pairing. If anything has gone wrong with the base pairing, it's able to go and do the correction. Now, as I told you, we call this polymerase 3 is the one which is laying down the new DNA strand. The building inspector, which is polymerase 1, is the one which is replacing the old primers, which is the RNA strands, pieces of RNA. That's the one which is doing. So with this, we conclude the concept of DNA replication. Now, if this kind of thing has to happen, it would take almost 1 month for it to become 2 new DNA strands. But now we can't wait for that length of period. So, see how nature has evolved and created newer concepts. What it does is, these are called as replication bubbles. So this opening, unzipping process will happen at different locations on the DNA. So these are called as replication bubble. Replication. It's not like it starts from one end, then it goes to the other end, all through right through. No. This replication bubble do occur at different intervals, and the unzipping can take place at different locations. It's like you've got a very long zip, and you can't start from one end of the zip to end up to this end. So people would have some kind of separations somewhere along the line. That is one very important thing that you need to understand. And this opening over here is called as a replication fork. It's called as a replication fork. So with this, we are completing the concept of DNA replication, and thank you for watching this video. And in the next video, we will talk about what is called as protein synthesis. Protein synthesis. And this is from Zambia. God bless.